All right, so once we found that DNA was the genetic material and we had evidence of DNA structure, then the next key thing was finding out how DNA was used to make proteins. And so uh, this was quickly, based on DNA structure, there were some very quick guesses that turned out to be quite right. That DNA was a code, and that code specified amino acids and proteins. And very quickly, biologists found that this turned out to be the same process in virtually all cells: prokaryote, eukaryote, plants, animals, everything. And the code turned out to be identical in all these cells, as well as the process. And the process was relatively simple. It was a two-step process. We now refer to those steps as transcription and translation. That virtually all organisms had a single enzyme called RNA polymerase that worked to make an RNA copy from a DNA template. And that RNA copy was then uh, converted into a protein sequence during translation, where ribosomes attached to the messenger RNA and linked together amino acids brought by transfer RNAs. And so we made protein from an RNA template. This code for the production of um, proteins, this involved three nucleotide sequences that specified a amino acid in a protein or an action to be taken, like a start or a stop. The genetic code stands for the sequence of mRNAs that are converted into a protein. The mRNA has three base sequences referred to as codons, and they are read by a three base sequence on a transfer RNA that brings amino acids there. You don't have to memorize the genetic code, but there it is. And so note that almost all proteins start with the amino acid methionine, which is coded for by the codon AUG, which is often referred to as the start codon. And then there are three stop codons. Transcription was first figured out in E. coli. And so prokaryotic transcription involved the enzyme RNA polymerase, which would recognize there was a piece of DNA to be copied by recognizing a specific sequence called a promoter, often referred to as the Tata box. And this would cause the messenger or the RNA polymerase to bind to the DNA and open the DNA sequence so that it could be copied. Okay, and so then complementary RNA bases are added to a growing strand that is based on the DNA template. Okay, the RNA, messenger RNA that's made begins usually with the codon AUG, and it will continue until there's a stop sequence that forms what's called a hairpin structure, and that causes the RNA polymerase to let go of the DNA and the transcription bubble to close. In prokaryotes, transcription is usually directly coupled to translation. So even before transcription is finished, the ribosome grabs onto the newly formed messenger RNA and starts making um, a protein from that. Okay, and usually um, in prokaryotes, related genes are all controlled by one promoter and they're all made together. And so these are referred to as operons. Transcription in eukaryotes is more complex. There are more RNA polymerases. Some of these are involved in making ribosomes. Some of them are more involved in making little RNA molecules called SNRPs that are involved in regulation of various processes. The RNA polymerases are larger than prokaryotic RNA polymerases. They have more subunits, and they require additional factors to get them active. They still bind to a promoter, usually the tau box, but now we need more different transcription factors. Eukaryotic transcription also begins with the promoter binding to the or the RNA polymerase binding to the promoter and with these transcription factors. And then the elongation also is by complementary bases being added to a growing RNA molecule, which is, of course, based on the DNA template. Again, there will be a stop kind of signal that terminates it. It's a little different than in other prokaryotes, but the resulting messenger RNA is not done. It's going to be processed. There are parts called introns or intervening sequences that are going to be cut out in a process called splicing. And this splicing can stick together the remaining parts called exons, which are going to be made into proteins. These exons can be put together in different ways. So one DNA molecule can be made into several different proteins. The way this is done is called splicing. And so these small RNAs are used to cut out these introns. These are called SNRPs, small nuclear ribonucleic proteins. And so they involve both RNAs and protein. These SNRPs can be cut out, and then there's additional processing with a cap added on the 5' end, and then a poly A tail added on the 3' end. These are signals for how much protein is going to be made, how long the messenger RNA is going to stick around, and they're also particularly the poly A tail is needed to get the messenger RNA out of the nucleus, because of course all the transcription occurs in the nucleus. What's going to happen then is that the sequence of bases on that messenger RNA is going to code for making a protein. And there are 20 different amino acids that can be assembled together in any order. They're different in size, but they all have a same basic structure with a central carbon that has an amino group, an NH3 on one side, and an acid group, a COOH group on the other side. And so proteins may have anywhere from 10 to several hundred amino acids linked together, and then they will fold up, and these can be then used for almost anything. I show the amino acids here. You don't have to know them or in any way, but know that all amino acids can be linked together easily because they have the same basic structure around the central carbon. And so in the box in the upper right, it shows a peptide bond, and it shows what happens is this is a dehydration, where a water unit is cut out and a nitrogen is linked to a carbon to form this structure. Okay, what brings the correct amino acids to be linked together are transfer RNAs, and there are more than 20 different types. On one end, they have an anticodon, which is going to pair up with the codons, the sequences on the messenger RNA. And then on the other end, they have an attachment site for RNA. Okay, and so in translation, what happens, and I'll go through first in the prokaryotes, there is a sequence on the beginning of each messenger RNA that includes an AUG. That's the start codon. And that's recognized by the transfer RNA that has an anticodon, UAC, and that's going to bring methionine. This will cause the small ribosomal subunit to attach, and then the large subunit will follow. And once that happens, then there is another site for the second codon, and it will have a site for a second transfer RNA, which recognizes the second codon, and will bring the second amino acid. The ribosome then catalyzes a peptide bond between those two amino acids and moves down the chain. So this is where initiation is complete. You can check a link there for the animation if you go to the PowerPoint. Okay, here's another view of the same thing. The Sol subunit, the there's the 5' end with the AUG at the near the start. Okay, an initiation factor binds to the transfer RNA carrying methionine. Once that happens, the large subunit attaches. Okay, then you get the elongation, which is where you're going to add amino acids, and so the ribosome will move down the messenger RNA, and as it does, new transfer RNAs will bring the correct amino acid, and there will be a peptide bond formed between the amino acid and the one on the growing chain, and this continues, it just keeps moving down. This will go on until termination happens, where there is a codon on the messenger RNA that doesn't correspond to any transfer RNA that brings an amino acid. Instead, it responds to, or it links to a release factor that recognizes that stop codon. And when a stop codon and the release factor are recognized, then the ribosomes will release the messenger RNA and the amino acid, and we're done. Key things here include the transfer RNAs that carry the amino acid, and these are attached by another set of enzymes that will attach it. And then the transfer RNAs have the anticodon loop. Okay, in eukaryotes, protein targeting may occur. Okay, translation in the cytoplasm or on the endoplasmic reticulum often includes extra kind of pieces of the protein that may be cut off later, and these will maybe have a sequence that allows the protein to go to a Golgi apparatus or another organelle. Eukaryotes make proteins more efficiently and have better mechanisms for targeting those proteins as to where they go and what they're going to do. And so these are related to these features like 5' caps and poly A tails and targeting sequences. With all these things, there's different ways to represent them. So I thought I'd show different ways that you see transfer RNAs there. Those all have anticodon areas that are recognizing the codons, and they all show an acceptor end. Here's another picture of the spliceosome, the SNRPs that form together to cut out the introns. Okay, well, we'll have more of this. I'll show some animations during class next time. This is the key thing all cells do. It's that all cells use the same process and same code is key for genetic engineering, and it was a complete surprise to biologists.